chapter 7.
Oh, 7.
Which is adjustments in physical relations. Okay? And what it does is it also looks at the what we call the reproductive systems. 1 Corinthians chapter 7 is a very important passage to study when you're busy with this. Okay? Because a lot of the arguments come from 1 Corinthians chapter 7. Obviously it will come from Ephesians chapter 5 and 6 as well. And then obviously Genesis. And there's other passages too, but that would be your main passages. Genesis 1 to 3, Ephesians 5 and 6, and then 1 Corinthians 7. Also obviously 1 Corinthians 13, which is the passage on love. One cannot describe that. Ephesians chapter 5 and 6. I think 5 is from verse 21 onwards. If I'm not mistaken, it's more towards the end. And then the beginning of chapter 6, then the last part of chapter 6 gives you the part on the clothing, warfare, and so forth. Or more prayer than anything else than warfare. It's more a case of clothing yourself with prayer than the warfare itself.
That's the armor piece that you get.
Yeah, let's not get mistaken.
And Genesis?
And Genesis 1 to 3.
Oh.
Yeah, that would be your main.
That's 1 to 3,
yeah.
so
you see,
chapter 1, chapter 2, and chapter 3.
Specifically in chapter 1 Corinthians chapter 7, it says, there's two biblical teachings there if you're reading verses 3 to 4. Let the husband fulfill his duty to his wife, and likewise also the wife to her husband. The wife does not have authority over her own body, but the husband does. And likewise, also the husband does not have authority over his own body, but the wife does. I remember when you're going into the marriage, some marriages doesn't start on an equal footing. Why? Because one of the people in the marriage might have had sexual relationships prior to the marriage. And it could be that they place strain on their partner to perform the way their previous partners has performed, now that they've gained experience, obviously. They might be shooting themselves in the foot and thinking that they are pretty good, and they may not be, to be very direct. So that's something else as well which they will have to go through in the marriage relationship. What this particular passage is saying is, and again, we need to be open about this. I'm thinking of husband is tired, but remember, husband is also very physical. He's drained and he's not feeling like anything. But his wife knows that he's physical. And so she will satisfy him sometimes. Simply because she knows it might give him a kick. It's those type of things that we need to discuss sometimes in the relationship very openly. Not that it's always a physical thing. Like I said to my friends on a particular occasion when we were having a discussion, we don't only have sex for the sake of reproduction. We also have sex simply to enjoy it. Enjoy it. Let's not fool ourselves. And sometimes it's just and sometimes we are selfish in that act. It's not, all things being equal, but they are not. So it's very important for you to guard your heart, your own heart in this matter, and to make sure that you don't deny your partner what is duly hers, or she denies you what is duly yours, and are unreasonable. But by the same token, don't be unreasonable towards your partner when they have had an excessively long day. And you've made no attempt on your part to connect with her emotionally, but now you want to connect with her physically. You may get yourself into trouble. You might get a good no from her. And for good measure, because women are different from us. And that emotional connection needs to be kept alive. Not for the sake of getting sex, but simply because you love her. And you want to care for her and look after her. Because if you're doing it simply for that, then you're still being selfish, aren't you? Well, that's my take on it. I think you're still being selfish. You're just doing it for the sake of that. And sometimes we are like that. We do it for an end result. And she will be asking, what do you want? But you must still agree, because there's a good relationship. You understand? Because you've set a platform for it. So you need to work also for it. If you don't work for things, they don't really matter, do they? If you get it on a platter, after a while it's a bit boring. In any case, so we're going to take a look at the reproductive systems quickly. First one is the female part, they're about 10 minutes each. You have not. So it would be, it's something that you need to raise with the couple, because there might be a possibility, or you may advise them to go and have a test. You can't tell them to have one. You may advise them to have a test, because they are looking one after the other. Remember, it's about caring for your partner. If you've had sex before, it would be advisable to go and have a test, because if it is the STIs and it can be dealt with, then it must be sorted out. If it can't be dealt with, in the case of HIV or AIDS, there needs to be open discussion. There are couples who are married, one who has the illness, but still the marriage is fruitful, and they are able to deal with it. So those type of discussions need to be even more open nowadays, especially with the fact that people are so loose in their morals. That discussion must take place in that environment. So don't be scared to raise it and then to advise if it is the case.
Maybe you've noticed that every time we talk about a new system, we highlight its importance by saying how you die without it. Like, without your muscular and skeletal systems, you'd collapse into an inert bag of goo. Or how, if we magically removed your respiratory or circulatory system, you'd die in a couple of minutes because your cells wouldn't have oxygen. That's because most of our body's systems are just trying to keep us alive minute to minute. But one of those systems doesn't really care if you live or die, at least not until it's done its job. It's how every living thing gets its start, but it doesn't really kick in until puberty, and even then, it's more concerned about investing in the future than keeping you alive. When it comes to your reproductive system, it's not concerned about you so much as it's concerned about your alleles, your genetic code, and the future of the human species, which are no small stakes. This system includes our primary internal sex organs, the gonads, like testes and ovaries, the various sex hormones they secrete, and the gametes, sperm and eggs, they produce. It also includes the glands, ducts, external genitalia, and particular brain parts that help the gonads and gametes do what they need to do, which basically is mate, combine alleles, and make babies. Now, all animals have their own particular and fascinating anatomical methods for getting their gametes together, and we could do a whole course just on that and never run out of material, and let me tell you, I would like to do that. But while we may seem kinda tame compared to animals that turn bright red or bite off penises or starve themselves for a chance to breed, our systems are still pretty complex. In fact, it's gonna take most of the month to get through all of our various anatomical parts and hormones and explain how sexy time, fertilization, pregnancy, and development work, starting today with the female anatomy. And remember, this is nothing to be shy about. When talking about sex, we are talking about the future of humanity. So when we talk about sex, we're talking about spreading our alleles around, but when we visualize sex, most of what we picture are our anatomies. Who has what and what goes where. In an anatomical female, that involves the vulva, which includes the mons pubis over the pubic bone and labia majora and labia minora, the elongated skin folds that surround the vestibule, which contains both the urethral and vaginal openings. Beyond that is the vagina, which I'm sure you know is how menstrual blood and babies leave the body and how sperm gets in. But as much as we tend to put all the focus on the bathing suit parts, those are only the genitalia, the external sexual organs. And they're really just a means of getting gametes together. Reproductively speaking, they're the least important parts of the system. The ground control of the female reproductive anatomy, the place where the orders are given, are of course the ovaries. Their main job is to produce and release female gametes and sex hormones like estrogen and progesterone. You'll remember from biology that gametes are haploid cells, meaning that they only have one set of chromosomes and are formed by meiosis. When a sperm fuses with an egg, they make a diploid cell, which has all the genetic genetic instructions required to make a baby, and pretty much everything about how our reproductive systems work is designed to make that happen. Each ovary lives inside a fibrous sac that consists of a layer of connective tissue called the tunica albuginea and another layer of cuboidal epithelial cells called the germinal epithelium, which is actually part of the peritoneum that lines the abdominal cavity. The ovary itself contains a cortex that houses developing eggs and a medulla that contains most of the ovary's blood vessels and nerves. But the business of passing on alleles and saving humanity really begins in the basic reproductive units in the cortex, the ovarian follicles. These are tiny sac-like structures that each hold a single primary oocyte, a sort of incomplete proto-egg, along with a bunch of supporting follicle cells around it. Females are born with essentially all of these early versions of eggs in all of the primordial follicles they will ever have, around 1 million at the time of birth. But right around birth, the oocytes stop developing. They get stuck at the first stage of meiosis, and they stay that way for years, sometimes forever. The actual process of egg creation, or oogenesis, is delayed until puberty when the rest of the body is physically ready to reproduce. Now this works differently for us than it does for some other animals. Like if you're a salmon or a mayfly, then all of your eggs will mature at once, and then you'll mate, reproduce, and usually die in quick succession. I mean, people talk about living fast and dying young, but that's too fast. So human eggs mature one by one, almost constantly, doled out so that every month or so a mature egg is either fertilized or dies to make way for a new egg. This should all sound familiar if you were born with female anatomy or know anyone who was, because it's a big part of the well-known monthly menstrual cycle. But the truth is, menstruation is only one part of one cycle. The menstrual cycle is what happens in the uterus to prepare for a fertilized egg. The other cycle, the ovarian cycle, is all about the maturation of the follicle and egg, and it's what actually drives the menstrual cycle. Every day, even before birth, a bunch of follicles will begin a process of maturation, very slowly morphing from primordial follicles into what's known as late tertiary follicles, which are the ones that will support a fully developed egg. This process takes 375 days. But out of that bunch of follicles, usually about 20 or so, only one follicle will end up supporting a single mature egg. The rest won't get the hormonal boost they need to bring the egg to completion. This is what happens to the ones that start maturing before puberty, for example, so they undergo atresia, a kind of programmed self-destruction. And because I keep mentioning puberty, which you've probably been through yourself, it should come as no surprise that all of this activity is regulated by sex hormones. Starting around puberty, the hypothalamus and pituitary set up two concurrent cycles: the ovarian cycle in the ovaries, which ripens eggs and secretes sex hormones, and the menstrual, or the uterine cycle, which prepares the uterus to capture and nourish any mature, fertilized eggs. When puberty begins, the hypothalamus starts up the ovarian cycle by secreting gonadotropin-releasing hormone about once a month. This is a sex hormone that stimulates the anterior pituitary to release two more hormones: follicle-stimulating hormone, you'll often hear it called FSH, and luteinizing hormone, or LH. The follicle-stimulating hormone lives up to its name by stimulating the growth of a follicle, but only one, the one that happens to be the furthest along in development at the time. The FSH drives that one lucky follicle to keep growing by triggering the follicle itself to secrete its own estrogen hormones, which locally signal the follicle to mature even more. That surge of follicle-secreted estrogen then ends up stimulating the pituitary to secrete another pulse of luteinizing hormone to finish the job. The LH gets to work on the oocyte that's been dormant inside the follicle and triggers it to finally start dividing again, getting it to complete meiosis 1 and move on to metaphase 2. This whole process takes about 14 days, at the end of which the follicle, which is now mature, pushes up against the ovary wall, ruptures, and with the help of enzymes breaches the wall and ejects a single, now mature oocyte. Congratulations! You've just ovulated! The damaged follicle now slows its estrogen production while morphing into a different structure called the corpus luteum, which eventually degenerates. But first, it releases its final hormonal swan song, a bunch of progesterone, a little bit of estrogen, and some inhibin that together stop the release of FSH and LH. They also prepare the uterus to receive the oocyte, which is now on its way down a fallopian tube where it might meet a nice young sperm. The tubes are about 10 centimeters long, and interestingly, they aren't actually connected to the ovaries. This means that when the egg pushes through the ovary, it has to float a short way through the peritoneal cavity before it's caught up by a fallopian tube. Now, only if and when an egg fuses with a sperm does it actually complete meiosis 2 and officially become an ovum. But whether it's fertilized or not, the egg works its way down the tube until it enters the uterus, a hollow, thick-walled, and very stretchable muscular organ that sits anterior to the rectum and posterior-superior to the bladder and ends with the cervix. And the uterine wall is composed of 3 layers: the perimetrium on the outside, the bulky, smooth muscle myometrium that contracts during labor, and the inner mucosal lining, the endometrium, which consists of a thin main deep basal layer and an outer functional layer. If fertilization does happen, then the new embryo snuggles into the endometrium for gestation, but the uterus is only receptive to implantation for a short time, about a week after ovulation. If the egg isn't fertilized, that outer functional layer sloughs off. And that's the first phase of the uterine or menstrual cycle, the series of changes that the endometrium goes through every 28 days or so in response to changing hormone levels and in coordination with the ovarian cycle. The shedding of the functional layer is triggered when the progesterone and estrogens that were being produced by the corpus luteum start to drop, about 10 days after ovulation. This phase lasts about 5 days. Meanwhile, the FSH and LH released from the anterior pituitary start to rise again, stimulating the next round of follicles which begin to make estrogen. This heralds the start of phase 2 of the menstrual cycle, the proliferative or pre-ovulatory phase, which typically lasts from days 6 to 14 of the cycle. The rising estrogen levels in the follicles stimulate the regeneration of the endometrium, building a cushy, well-vascularized habitat for another potential fertilized egg to call home. And after the next egg is released, the final secretory or post-ovulatory phase begins. This is when the ruptured follicle forms in the corpus luteum, and if fertilization didn't happen, the corpus will stop producing progesterone and the endometrium will start to shed its functional layer and it starts all over again. But if by this time the egg has met a nice sperm and gotten fertilized, then the pulse of progesterone from the corpus triggers even more thickening of the functional layer of the endometrium and a secretion of nutrients that will tide an embryo over until it has implanted itself in the blood-rich lining. Which is a big if. Like its whole separate video big, so that's where we're gonna go next time. But for now, you learned all about female reproductive anatomy, how sex hormones affect oogenesis and ovulation, and how the ovarian and menstrual cycles mature and release oocytes and create a comfy uterine environment for a fertilized egg. Thank you to our headmaster of learning, Linnea Poyev, and thank you to all of our Patreon patrons whose monthly contributions help make Crash Course exist not only for themselves, but for everyone everywhere. If you like Crash Course and want to help us keep making videos like this one, you can go to patreon.com/crashcourse. This episode of Crash Course was filmed in the Dr. Cheryl C. Kinney Crash Course studio. It was written by Kathleen Yale, edited by Blake DiPastino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins, edited by Nicole Sweeney, our sound designer is Michael Aranda, and the graphics team is Thought Cafe. Sex is a big gamble. I mean, the mental risks alone that we take with our relationships are taxing enough. When is he gonna text me back? Am I good enough? What if she doesn't like the real me? But the emotional price we pay for intimacy can almost seem paltry compared to the physical investments that our bodies make in order to reproduce. And the sexes each make this wager differently. The female gamete is, biologically speaking, a big bet. It's like putting all your chips on black. An ovum and a follicle take over a year to develop, and as human cells go, it's huge, about 1/10 of a millimeter, just large enough to be seen with the naked eye. Plus, it requires a whole network of supporting tissues in the uterus while it waits for fertilization, which, if that doesn't happen, then have to be shed and rebuilt a month later. And then there's the commitment. Once an egg has been ovulated, everything in the female reproductive system is dedicated to dealing with that one cell for almost a month. And if it gets fertilized, you're looking at 10 months. But when it comes to playing the reproductive roulette wheel, the male reproductive system takes the opposite strategy. If female gametes amount to a high-stakes bet, the males are basically the penny slots. Human sperm are only about 100,000th the mass of an egg, and they're almost as easy to produce as skin cells. Actually, the process by which they mature is pretty similar. That's because sperm are small and stripped down, not invested in much beyond a nucleus and a tail and some extra mitochondria to fuel the journey. All they have to do is drop their DNA and fall apart. The risk of failure is still there. In fact, with male gametes, the odds are overwhelmingly against any single sperm actually accomplishing its mission. But while the female reproductive system meets this risk head-on with a single big play, the male's tries to beat the odds with sheer force of numbers. Here's a question for you. Why are the most important parts of the male's reproductive system literally left hanging? The testes, the gonads of the anatomical male, are tasked with making male gametes, sperm, and the androgen hormone testosterone. But unlike the ovaries, which are protected inside the body, testicles dangle outside the abdominal cavity in the scrotum. Because, even though sperm may not be terribly big or complex, they're touchy when it comes to temperature. The rate at which they divide, copy DNA, and even feel the effects of some hormones are all diminished at the core body temperature of 37 degrees. So the testes have to be outside the body cavity to reach the lower temperature necessary for proper spermatogenesis, or sperm production. It's a rare instance of sensitivity among what are otherwise pretty hardy and expendable cells. But that's not to say that the testes aren't complex. Each one is divided into about 250 sections called lobules, which are loaded with tightly coiled seminiferous tubules. These tubules are the sperm factories, made of a stratified epithelium surrounding a central fluid-filled lumen. And this sperm-making work is supported by cells throughout and around the tubules. Among the most important are the Sertoli cells inside the tubes, which nourish developing sperm cells, sort of like how female follicle cells help oocytes. There are also Leydig cells that secrete testosterone, much like the corpus luteum that secretes estrogen. But compared to egg making, spermatogenesis is, as I mentioned, a pretty cheap and easy process biologically. It's kind of like how your skin cells are produced, with stem cells at the basal layer that produce immature cells, which in turn get forced toward the surface as they mature. But in this case, the sperm develop on the outer edge of the tubule and progress inward toward the lumen. And it all begins with a trigger that, if you've seen our lesson on female reproduction, should look pretty familiar: a hormonal cascade. When puberty comes a-knockin', the hypothalamus starts releasing gonadotropin-releasing hormone, or GnRH, and this tells the anterior pituitary to secrete follicle-stimulating hormone and luteinizing hormone into the blood, just like in females. But in females, LH leads to the release of estrogen in the follicles, whereas in males, it spurs the Leydig cells to release testosterone. Meanwhile, the FSH triggers the Sertoli cells to release androgen binding protein, or ABP. This binds to the testosterone, creating large local concentrations of the stuff, which is ultimately what triggers the production of sperm. The targets of the testosterone are the outermost cells in the tubules, called spermatogonia. They're the stem cells that set the sperm-making process in motion by dividing. And actually, they've been doing that all along, even before puberty. Throughout childhood, a spermatogonia cell uses mitosis to continually divide into two identical daughter cells. So when puberty starts, the testosterone causes them to divide differently. Instead of splitting into two identical spermatogonia cells, they begin producing two distinct spermatogonia. One type of daughter cell, known as a type A cell, stays up near the basal lamina and just keeps dividing, so you never run out of spermatogonia. But the other kind, the type Bs, get pushed down the tubule toward the lumen and turn into primary spermatocytes. These primary spermatocytes then go into meiosis I and form two smaller haploid cells called secondary spermatocytes. They then rush through meiosis II, and their resulting daughter cells total 4 round spermatids. These spermatids now have all of the 23 chromosomes they need for fertilization, but they aren't exactly mobile. If they're ever gonna find themselves an egg, they need a way to get around. They need a tail. The process by which a spermatid elongates, grows a tail or flagellum, and officially becomes a mobile sperm is called spermiogenesis. And the whole process takes about 5 weeks, but it's not like it holds anything up, because there are plenty more where those spermatids came from. In the end, each primary spermatocyte gives rise to 4 actual sperm, and considering how many spermatogonia are continuously dividing into spermatocytes, it's easy to see how a mature male can crank out 1,500 sperm a second. That's a far cry from all the investment that goes into making a single ovum. Now, obviously, if sperm are gonna get anything accomplished, they have to leave the seminiferous tubules that made them, so even once they have tails, they still need a little help getting going. That's why each tubule is surrounded by several layers of myoid cells, which, like smooth muscle, rhythmically contract using peristalsis to squeeze sperm and some fluid secreted by the Sertoli cells through all the twists and turns toward their next destination, the rete testis in the posterior testis. From there, the sperm, although still immobile, leave the testes by way of the epididymis, a long tangled set of tubes behind the testes where they'll spend the next few weeks gaining their mobility. The bulk of the epididymis consists of the enormous duct of the epididymis, which, believe it or not, could be uncoiled to stretch about 6 meters. Though I don't suggest doing that. This duct is full of tiny microvilli called stereocilia, which provide a huge surface area to help reabsorb some extra fluid and help pass along nutrients to feed the idling sperm. It takes sperm nearly 20 days to work their way through this labyrinth, during which time they continue to mature. Once through the duct, they enter the inferior epididymis, where they gain mitochondria, so they'll have energy to swim hard at a moment's notice. At this point, they have all the hardware to swim, but still lack the ability to actually move on their own own, a skill kept under wraps until they leave the epididymis and get activated by a series of glandular secretions. When that time comes, during ejaculation, the sperm flow from the epididymis through the vas deferens, a tube that travels up behind the bladder and joins with the duct from the seminal gland to create the ejaculatory duct. The left and right ejaculatory ducts pass into the prostate gland, where they empty into the urethra, which runs from the bladder through the penis and into the outside world. This system of tubes feeding into tubes allows all of the necessary glands to make their contributions to a moving wave of seminal fluid that helps sperm mature and perform their ultimate function, fertilization. The resulting mix of sperm, testicular fluid, and gland secretions, which we call semen, provides sperm with transportation, nutritional energy, chemical protection, and finally activates their motility. So let's take a look at these glands. The seminal vesicles are a pair of small, hollow glands behind the bladder that secrete a yellowish, slightly alkaline fluid that contains coagulating enzymes, fructose, and other things to help nourish and transport sperm. Interestingly, the vesicles also secrete special prostaglandins that help increase sperm success outside the body by decreasing the viscosity of a female's cervical mucus and actually triggering a reverse peristalsis of the uterus that helps draw sperm up the female reproductive tract. Now, the prostate gland encircles the urethra just inferior to the bladder. During ejaculation, it contracts to squeeze its own special secretion into the urethra. This stuff contains mainly citric acid and an enzyme cocktail that helps keep semen liquefied and thus easier to move and swim through. And finally, we have the pea-sized bulbourethral glands below the prostate that secrete a clear mucus that drains into the urethra to clear out any acidic urine prior to ejaculation. Speaking of, by now you may be wondering if we're ever gonna get to what's probably the first thing you think of when it comes to the male anatomy. The penis hangs from the perineum, between the pubic bone and coccyx. It consists of a shaft that ends in an enlarged tip called the glans penis, surrounded by a fleshy cuff of foreskin. Internally, the penis contains 3 layers of erectile tissues, each wrapped in a layer of dense, fibrous connective tissue. This spongy network is made of connective tissue and smooth muscle that's full of tiny vascular spaces that fill with blood during sexual arousal and make the penis expand and become rigid. The resulting erection is what helps the penis penetrate the vagina. And the fact that we're only now getting around to talking about it here at the end should tell you that, as with the female external genitalia, the penis isn't actually all that important in terms of overall reproductive function. Evolutionarily speaking, it's just a delivery system designed to get male gametes as close as possible to female gametes and reap some payoff for their investments. Now, what that payoff looks like in terms of fertilization is where we're headed next week. But for now, you learned probably all you'd ever care to know about testicular anatomy, the steps of sperm production, and how it's influenced by gonadotropin and testosterone. We also looked at how sperm mature and how they leave the body on a tide of glandular secretions during ejaculation. Thank you to our headmaster of learning, Linnea Boyev, and thank you to all of our Patreon patrons whose monthly contributions help make Crash Course possible, not only for themselves, but for everyone everywhere. If you like Crash Course and want to help us keep making videos like this one, please check out patreon.com/crashcourse. This episode was filmed in the Dr. Cheryl C. Kinney Crash Course studio. It was written by Kathleen Yale, edited by Blake DiPastino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins, edited by Nicole Sweeney, our sound designer is Michael Aranda, and the graphics team is Thought Cafe. Welcome to the main event. After weeks of talking about our reproductive parts and how they work, today we finally discuss what happens when those parts come together to do what they were born to do, the sex. But first, let's talk about what we won't be talking about. We're not going to give any advice. We're not going to talk about STIs, even though some STIs can affect conception. We're also not going to talk about sexual behavior or mate selection, and there will be no discussion of birds or bees. And while people have sex in lots of different ways and for lots of different reasons, we're only going to be talking about the kind of sex that could lead to fertilization. We are going to talk about contraception, though, in terms of how and where and when different kinds can prevent fertilization. And we're going to look at what exactly is going on in your body before, during, and after sex, in the most very safe for work clinical terms possible. Humans have been experiencing coitus for as long as they could be called humans, and actually before that, because obviously there would be no humans without the sex. And although it's something that generally comes pretty naturally, it wasn't until the late 1950s and 60s that anyone thought to look at the physiological science behind the act of sex. So when American gynecologist William Masters and sexologist Virginia Johnson kicked off their unprecedented and controversial research into the so-called human sexual response cycle, they began by inviting hundreds of male and female volunteers to come into the lab and have sex in the name of science. Though it wasn't as voyeuristic as it might sound. Outfitted with loads of wires and heart monitors, volunteers were periodically interrupted in the middle of the act to be measured. 10,000 trials or so later, Masters and Johnson found out some interesting stuff. Namely, that a typical complete sexual cycle runs through 4 distinct phases: excitement, plateau, orgasm, and resolution. During the excitement phase, things are just getting going. Sexual arousal triggers the parasympathetic reflex that causes arterioles in the external genitalia to dilate and fill with blood, which is actually one of the only times the parasympathetic system controls any arterial action. The plateau phase is marked by increased muscle tension, heart rate, blood pressure, and breathing rates. Here, the male urethral sphincter contracts to make sure urine doesn't mix with semen. During orgasm, pelvic and other muscles around the body contract rhythmically as an anatomical male ejaculates, releasing the sperm that might just get a chance to fertilize an egg, depending on the situation. Female orgasm is not required for conception, and some may never experience one, but if it does happen, the resulting uterine contractions and increased lubrication can help draw up and hold sperm in the uterus. As the body relaxes after all that excitement, it enters the resolution phase. Here we see males entering a refractory period that may last several minutes to a day or two, during which they are usually unable to reach orgasm again. Anatomical females are thought to not need this time out physiologically, though they may need it physically. Instead, some females are capable of serial orgasms one after the other. Masters and Johnson's works has its share of critics, many of whom point out that not everyone fits into their 4-stage mold, and that things are not always quite so linear in the heat of the moment. But overall, their core physiological framework is still accepted. Now, regardless of exactly how sex happens, if it involves a fertile anatomical female and male and the timing is right, it could ultimately end with fertilization. So how do a single little ovum and an even tinier sperm come together to make a fertilized egg or zygote that divides and multiplies and grows into the trillions of cells that make up the glorious, complex individual that is you? You'll remember that during ovulation, about once a month, an ovary releases a secondary oocyte that heads down the fallopian tube and toward the uterus. That oocyte has a window of about a day or less to meet a sperm before it's no longer viable. But that being said, sperm can persist in the female reproductive tract for as much as 3 to 5 days. Which means for fertilization to occur, they have to either be in place during ovulation, or they have to arrive no more than a day after. But now I feel like I'm getting a little ahead of myself. Let's go back to how sperm typically get into the anatomical female body in the first place. During sex, if a male ejaculates into a female's vaginal canal, millions of hopeful sperm will be on their way. Despite the fact that they only have about a dozen or so centimeters to go, their journey isn't for the faint of heart. Luckily, they don't have hearts. The truth, of course, is that most sperm never reach an oocyte. Some just go in the wrong direction, others are killed in the strange new acidic environment of the vaginal canal, and millions more fail to penetrate the gauntlet of mucus that surrounds the cervix. Even the ones that swim hard enough in the right direction and manage to reach the uterus may then be culled by defending resident white blood cells. In fact, sometimes only a dozen or so of the original millions make it through all those trials to actually reach an oocyte. But even if they do, it's still not over for them. The sperm that do make it to the uterus and then the fallopian tube have another hurdle to overcome because they can't actually penetrate an oocyte. Not yet. They could literally hang out right next to one for hours and be unable to make a move until they are what's known as capacitated and go through one last set of changes. As they swim through the cervix, uterus, and fallopian tube, female secretions start to degrade some of the sperm's protective proteins until the cap on the tip of its little head, called the acrosome, is fragile enough that it can leak special hydrolytic enzymes. Those enzymes are the key to getting through the oocyte's protective outer layers. But before that, it has to weave through the granulosa cells that form the oocyte's outermost layer, known as the corona radiata. Once it's past that, it runs into the zona pellucida, a layer of glycoprotein that covers the oocyte's plasma membrane. And this is where it starts to get a warmer reception. This layer has specific sperm receptors waiting for it to bind to, and when it does, it opens a bunch of calcium channels which flood the sperm with calcium ions. These ions trigger the final acrosomal reaction that releases the enzymes the sperm needs to work through the zona pellucida. Now, a number of sperm make make it to this phase, and in terms of strategy or luck, it's best to arrive on the scene after your compadres have already been hard at work digesting holes through the pellucida. Because once a path is cleared, a lucky sperm can wiggle against the now exposed oocyte membrane until it locks onto another set of sperm-binding receptors. And when a sperm docks into one of those, the membrane of the sperm fuses with the innermost membrane of the oocyte, and the contents of the sperm enter the oocyte proper. Then, something pretty cool happens. Remember that the egg is still just a secondary oocyte here and hasn't completed meiosis. As the sperm delivers its payload, it causes a flash of calcium ions from the oocyte's endoplasmic reticulum. This tells the secondary oocyte to get ready to complete its second meiotic division, but it also triggers the reaction that suddenly seals the egg by destroying all the leftover sperm receptors in the membrane, preventing any other sperm from weaseling in. The oocyte then quickly completes meiosis too, leaving two daughter cells, the large ovum and a puny second polar body. Meanwhile, the sperm's detached nucleus swells up to 5 times its original size, forming the male pronucleus, one of 2 sort of protonuclei that contain each gamete's genetic information. The other one, the female pronucleus, takes shape from the nucleus of the ovum, and then the two start to approach each other, pulled along a protein scaffolding or mitotic spindle between the two. When the membranes of the 2 pronuclei rupture, chromosomes go flying, and boom, you officially have a diploid zygote, which almost immediately starts mitosis. And that, folks, is how you fertilize an egg. But even though our bodies are made to have lots of sex and babies, there are plenty of people who are interested in enjoying the sex part without enjoying the baby part. And given all the things that usually need to go right to achieve fertilization and implantation, a little disruption at some stage along the way is usually all you need to remain baby-free. So folks have been experimenting with various herbal, chemical, physical, and behavioral methods of contraception for centuries, for millennia. Some of which have been pretty nutty, like drinking cocktails laced with lead, or wearing weasel testicle amulets and rabbit butts, or shoving herbs in places where herbs should not go. But of course, contraception has come a long way in recent years. In cultures that provide access to it, folks have a wide range of contraception options, in addition to abstinence. First, if you're really sure you don't want kids, or more kids, sterilization is an option. During a tubal ligation or vasectomy procedure, a doctor snips, blocks, burns, or ties up the fallopian tubes or vas deferens, effectively ending the gamete express by permanently preventing the sperm and egg from ever getting to the penis or uterus. But those who prefer a less permanent option also have lots of choices. We've got barrier methods that work mostly by physically keeping sperm and egg on either side of a wall, which is usually made of latex. Condoms, of course, cover the penis and catch ejaculated sperm before it can go anywhere, while diaphragms, sponges, cervical caps, and female condoms are inserted into the vagina, where they can form a wall over the cervix, turning sperm away. Hormonal methods of contraception, like the pill or any number of newer shots, patches, and rings, use synthetic estrogen and progestin to prevent the progression of the ovarian cycle to ovulation, and sometimes make changes to the functional uterine layer, the endometrium, and cervical mucus to help deter sperm. An IUD, or intrauterine device, is a small T-shaped object a medical provider inserts into the uterus to prevent zygotes from implanting. And the implantation is key, because even if a sperm and egg manage to complete fertilization, in order for gestation to take place, the zygote has to find a home for itself in the uterus pretty quickly so it gets the nutrients it needs to continue developing. So that's what we're going to be talking about next time. It takes a long time to talk about all the ways that we make a baby, but it's important. But for now, you learned about the 4 phases of the human sexual response, how a sperm finds and fertilizes an egg, creating a zygote, and how different types of contraception work to prevent that from happening. Thank you to our headmaster of learning, Linnea Boyev, and thank you to all of our Patreon patrons whose monthly contributions help make Crash Course possible not only for themselves, but for everybody everywhere. If you like Crash Course and you want to help us keep making videos like this one, you can go to patreon.com/crashcourse. This episode was filmed in the Dr. Cheryl C. Kinney Crash Course studio. It was written by Kathleen Yale. The script was edited by Blake DiPestino. Our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins, edited by Nicole Sweeney. Our sound designer is Michael Ronda, and the graphics team is Thought Cafe. You were once the size of the head of a pin. Let's just start out with that. 2 millimeters, at the most. And today, I don't know you, but I'm assuming that you're a lot larger than that. Many, many pinheads from head to toe. Assuming the average human is about 1.7 meters, it's safe to say that you're about 850 times taller than when you started out as a zygote. I think we all deserve a round of applause. I'm very proud of us. But how did it happen? How does a single cell eventually grow into the fully formed several trillion-celled body we've spent all year discussing? And for that matter, how the heck does a female body nurture, protect, and generally tolerate growing a whole new person inside of it, with all the mood swings and sore and swollen parts, increased blood volume, constant pee breaks, and general body weirdness? On an emotional level, I have no idea how the mothers, or the zygotes for that matter, endure all of that. But on a physiological level, like so many aspects of our body's functions, pregnancy begins and ends with the same thing: hormones. Last week we left off with a sperm and an egg against all odds finally getting together to make a zygote. Now, how that tiny head of a pin gets to be even a fraction of the size you are now, a newborn human that's say 50 centimeters long, is complex. It involves a system of hormonal signals that, as sophisticated as they are, get interpreted by the very earliest human cells into 3 simple instructions: divide, differentiate, develop. The first step, dividing, begins in what's known as the cleavage phase, when cells cleave, or split in two, over and over. It starts about 24 hours after fertilization, when a little zygote turns from 1 cell into 16 cells called blastomeres. The cells divide so quickly that they don't actually grow between divisions, they just create more smaller cells. This allows each little cell to have more surface area, which helps them take in the oxygen and nutrients they need from their environment. And of course, it also creates more raw materials for building an even larger zygote and eventually an embryonic human. Like, you don't make a car by carving it from a single chunk of metal. You put together lots of smaller components, each with its own form and function. That's what these cells are during cleavage. About 3 days after fertilization, these divisions have formed a little berry-shaped cluster of cells that looks different and complex enough to get a whole new name: a morula, from the Latin for mulberry. It's one of the cuter terms you'll come across in human physiology, and it also marks the end of the cleavage stage. Because the little cells that make up the morula don't stay together as a solid mass. Instead, they start to form a hollow sphere filled with fluid, a blastocyst. A blastocyst contains a single outer layer of large, flat trophoblast cells, and inside, a cluster of smaller cells forms called the inner cell mass. This mass is what's going to turn into the embryo eventually, while the trophoblasts will go on to form the placenta and blood vessels that will nourish it. So keep in mind that during all these early divisions, the zygote and then the morula are on the move. They're headed down the fallopian tube toward the uterus. So in addition to manufacturing a ton of tiny pieces and assembling them into something that's getting increasingly complex, the whole operation is mobile. It's kind of like you're building a car while you're driving it. When the blastocyst reaches the uterus, it just floats around for a few days, soaking up secretions that are full of vitamins and glycoproteins, looking for a place to call home. But soon it's going to need permanent nutritional support. So about a week after ovulation, it snuggles up to the endometrial layer and starts the process of implantation. Now, up to this point, we've just been talking about the zygote itself and the changes it's been undergoing, and we're all very proud of it. But from here on, the changes are best understood not just in terms of the budding, soon-to-be embryo, but in terms of the whole environment where its changes are taking place. That environment being the mother's body. Implantation is only possible, after all, thanks to surges of estrogen and progesterone from the corpus luteum, the ruptured follicle that we talked about last time. And together they prepare the endometrium to receive the blastocyst, allowing the uterine lining to bind to little proteins on the trophoblasts, holding onto them for the duration of the pregnancy. If all goes according to plan, implantation takes about 5 days and finishes up around 12 days after ovulation, right around when menstruation would otherwise kick in and slough off the endometrium. If that happened, it would be bad, so trophoblasts secrete a luteinizing-like hormone called human chorionic gonadotropin, or HCG. This hormone bypasses the whole hypothalamic pituitary ovarian axis, if you remember that, and talks directly to the corpus luteum, telling it to keep pumping out estrogen and progesterone. So the first thing to notice here is that from this point on, most of what's happening in this little clump of cells is happening because hormones. HCG is basically calling all the shots, triggering the release of other hormones that are crucial to the blastocyst's development. But while hormones are doing the grunt work, you might also notice that the blastocyst is really in charge now. Essentially, it has taken over hormonal control of the whole uterus. The release of hormones is that powerful. In fact, a whole new, albeit temporary, organ is forming at this point too, which is going to take over the hormone-controlling job. That's the placenta. The placenta is one of the defining and most amazing structures in the mammalian class. It's an organ that only appears during pregnancy and is created by the melding of both maternal and embryonic tissues. Together with the umbilical cord, it provides for the direct transfer of nutrients, hormones, and wastes between mother and offspring. These elements start to really take shape shape after implantation is complete, and we enter the embryonic stage. This is where the blastocyst differentiates into various cell types and develops into a legit embryo surrounded by an amniotic sac and hooked up to the placenta. And around the end of week 8, this tiny thing is now officially a fetus. Over the next several months, it rapidly develops organ systems and bones and grows into what you see in the delivery room. But rather than focus on those changes, I want to look more at what profound and frankly sometimes bizarre adaptations the mother is going through. The most obvious changes are, of course, the anatomical changes. Basically, everything's getting huge. The mother's breasts swell and engorge with blood, and her baby bump gains dimension as the normally fist-sized uterus expands, pushing organs out of the way until it pretty much takes up the entire abdominal cavity from the diaphragm to the bladder. The growing placenta is still secreting estrogen and progesterone, but it's also pumping out relaxin, a hormone that loosens joints and ligaments to increase flexibility, and it also releases human placental lactogen, or HPL. This hormone cocktail does helpful stuff like tell the fetus to grow and get the breasts ready to lactate, and it also causes the mother's body to start hoarding glucose for the fetus to use. This increase in metabolism, combined with the fact that the kidneys also have to process waste from the fetus, leads to greater urine production, which every expectant mother is familiar with. It also has a huge effect on the cardiovascular system, because a pregnant woman's blood volume can increase by as much as 40%. 40%! Imagine walking around with an extra 2-liter bottle's worth of blood in your body and how much harder your heart would have to work to move it all around. This increased blood flow and pressure can actually make your gums swell and bleed, and fluid retention can literally change the shape of your corneas, potentially blurring your vision. Not only that, but the expanded uterus compresses pelvic blood vessels, affecting veins' ability to bring blood up from the lower limbs, resulting in swelling varicose veins, and if all that weren't bad enough, hemorrhoids. Fortunately, every pregnancy eventually comes to an end, usually about 38 to 40 weeks after fertilization, if all goes as planned. But unfortunately, the process of getting the baby out is pretty much a painful mess. Ask any mother. Giving birth is not for the faint of heart. The process of preparing the body for labor and then actually initiating it begins again with a change in hormones. Up until the last few weeks before birth, the placenta has been kicking out equally high amounts of both progesterone and estrogen. One of progesterone's main jobs has been to keep the smooth muscles in the uterus relaxed so they can't contract and stimulate labor too early. But as the due date nears, the mother undergoes a sudden decline in progesterone. Now, estrogen takes over. This is partially because the fetus itself is ready to go, and it starts releasing hormones like cortisol that tell the placenta to release even more estrogen to get the uterus ready for birth. Just like HCG calls the shots around the time of implantation, here estrogen is barking out all kinds of orders. For one thing, it prepares the uterus to start receiving new chemical signals by triggering its myometrial cells to start making receptors for the hormone oxytocin. It also triggers the formation of gap junctions between smooth muscle cells in the uterus. This will let individual muscle cells contract simultaneously when the time comes. Then, as labor nears, special cells in the fetus itself start secreting oxytocin, which binds to all the newly minted receptors and tells the placenta to release prostaglandins. Together, both of these hormones, oxytocin and prostaglandins, stimulate the uterine muscles to start contracting. When the contractions get strong enough to distend the cervix, it stimulates the release of even more oxytocin and prostaglandins, which keep the contractions rolling in one big positive feedback loop, initiating labor. The earliest stage of labor, dilation, is the period from when the first contractions begin to when the cervix becomes fully dilated to about 10 centimeters. During this time, each new contraction pushes the infant's head against the cervix, causing the cervix to thin and dilate. Once the cervix is fully dilated, the mother should feel the urge to push. The resulting expulsion stage lasts from full dilation through crowning and actual delivery as the infant is pushed headfirst through the cervix and out the vagina. But even after the baby is out, the mother still has a placenta to get out. Within about 30 minutes of delivery, strong contractions carry out the placental stage of labor, dislodging the placenta from the uterine wall to deliver the so-called afterbirth. Then finally, the mother can rest and marvel at how her body just built another tiny body inside it, complete with all the complex systems we've spent the last year talking about. And maybe someday that little baby will grow up and get a twinkle in its eye and combine alleles with somebody else and start the whole process all over again. And that is how the human race continues to exist. Today, you learned about the stages of pregnancy, beginning with how a zygote develops into blastomeres, to a morula and a blastocyst, and finally to an embryo and a fetus. You also learned about the amazing anatomical changes that take place in the mother and the hormonal sequence of events that lead to labor. After that, all you have to worry about is how you're gonna put the little thing through college. Thank you to our headmaster of learning, Linnea Boyev, and thanks to all of our Patreon patrons whose monthly contributions help make Crash Course possible, not only for themselves, but for everyone everywhere. If you like Crash Course and want to help us keep making videos like this one, you can go to patreon.com/crashcourse. This episode was filmed in the Dr. Cheryl C. Kinney Crash Course studio. It was written by Kathleen Yale. The script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins, edited by Nicole Sweeney. Our sound designer is Michael Aranda, and the graphics team is Thought Cafe.